Antibodies. Everyone has them. They're inside you right now. Millions of them, maybe even billions. Some scientists even estimate you could make a quintillion potential different antibodies. But what are antibodies? How do they work? And why do we care so much about them? Antibodies are Y-shaped proteins found throughout the body that bind specifically to a particular substance. They're approximately 150 kilodaltons in size and are sometimes referred to as immunoglobulins. During an immune response, their production is increased. Their job is to find foreign substances, like pathogens, so they can alert the immune system to wipe them out. They do this by identifying and interacting with antigens with exquisite specificity. An antigen is a molecular structure that an antibody recognizes, usually found on the surfaces of those foreign bodies. The specific part of the antigen that the antibody binds to is called the epitope. Antigens are biomolecules, usually proteins, doing any number of jobs. For example, the proteins that make up a virus's outer shell. Or the proteins that a virus uses to infect cells. Antibodies are produced by B cells, immune cells that originate in the bone marrow. When a naive B cell senses that it has found a new antigen, it gets activated, proliferates, and undergoes an extensive maturation process. This results in the production of highly specialized and specific antibodies. Once these antibodies are secreted by B cells, they circulate in the body. When they encounter their antigen, they bind to it and induce a specific immune response. But how do they do this? For antibodies, function follows form. So to understand an antibody's work, we first need to understand its structure. Each antibody is made up of 4 polypeptides, 2 identical light chains, and 2 identical heavy chains. These polypeptide chains come together to form that Y-shaped structure we mentioned earlier. The top portion of the Y is the fragment antigen binding region, or Fab, which binds to, you guessed it, antigens. The stem of the Y is the fragment crystallizable region, or FC, which triggers the immune response. Ready to get up close and personal with the Fab region? Fabulous. The tips of the Fab are known as the paratopes. These are the sections that identify and bind to an antigen. This is also known as the variable region of the antibody, since these sections can better change to fit an antigen. Part of the DNA that codes for this region hypermutates during the B-cell maturation process. This process is a bit of trial and error, with different B-cells making slightly different copies, and comparing results to see which copy fits the antigen best. This allows for a huge variability in the binding region. Paratopes can identify the antigen by binding to epitopes, which are also short, usually between 4 and 12 amino acids long. Remember that exquisite specificity we talked about? Paratope-Epitope interactions, the binding we mentioned, are the reason antibodies are so specific. The 2 topes fit together like a jigsaw puzzle. Each paratope tightly binds to one specific epitope. Of course, proteins are big, much larger than the short epitope that a paratope recognizes. But this means you could have antibodies specific to different epitopes, all part of the same antigen. Or you could have multiple antibodies that recognize the same epitope in slightly different orientations. It's important to know that the antibody binding strength can be measured by affinity, the strength of the binding between a single paratope and a single epitope. and by avidity, the combined binding strength of all paratope-epitope interactions. So even if an individual paratope has low affinity, the whole complex could have high avidity. But how does the antibody work with other molecules? That's where the FC comes into play. Fragment crystallizable doesn't exactly sound like a protein function. It sounds more like a Sailor Moon move. And it doesn't explain what the FC region does. That's because the name comes from an observation by English biochemist Rodney Porter, who noticed that this region of the antibody crystallized in his refrigerator overnight. He, and Gerald Edelman, eventually won a Nobel Prize for their antibody research and discoveries. So, why is understanding the FC so important? Unlike the Fab, which binds to the pathogen, the FC can bind to other components of the immune system, triggering different immune responses. These immune responses can be innate, with fast deployment of immune cells like natural killer cells, and broad defense mechanisms like fever, or adaptive. Adaptive immune responses include development and deployment of antibodies and antigen-specific T cells. In mammals, there are 5 isotypes, or classes, of immunoglobulin FCs, which are encoded by different heavy chains: gamma, mu, alpha, delta, and epsilon. Even though B cells only produce one type of antibody, defined by its binding region, it can produce different isotypes of the same antibody through a process called class switching. In an immune response, the first isotype to develop is IgM. They often show up on the scene 5 to 7 days after an initial exposure. Once secreted, they become pentamers, or 5 antibodies connected by a joining chain. Typically, binding strength of an IgM paratope is relatively low. But it has strength in numbers with its 10 binding sites, meaning that IgM has high avidity that makes up for its low affinity. Once IgM binds, forming an antigen-antibody complex, the FC region can activate a specific frontline immune response called the complement system. This response can destroy pathogenic bacteria and clean up damaged cells. Another isotype, IgG, is a monomer that is fairly common in early immune responses. IgG is generally found on the surface of B cells, or free-floating in your blood serum, and can act in several different ways. IgG can physically interfere through agglutination, which forces the antigens to clump together. Or they can use FC interactions to recruit other members of the immune system to attack the pathogen. After binding, IgG can also trigger opsonization, an immune signaling cascade that results in a response by a class of white blood cells known as phagocytes. Another defensive mechanism used by IgG antibodies is neutralization, effectively blocking the epitope a pathogen would have used to infect a cell by binding to it first. Next up, we have IgA antibodies. Once secreted from B cells, they form a dimer with 4 binding sites. Once created, they're considered the body's first line of defense. You can find IgA antibodies in the respiratory tract and other mucosal sites. While IgG is the most abundant antibody in the blood serum, IgA is the most abundant antibody throughout the entire body, due in part to its presence in mucosal secretions. These antibodies prevent colonization by triggering cells to create a surface coating. Imagine them helping to slime your cells, triggering them to secrete a layer of mucus that can stop pathogens from binding. Since they're found in secretions, they're also found in breast milk, providing infants with what's known as passive immunity. Next up, we have IgD antibodies. They're found almost exclusively on the surface of B cells. They're one of the least abundant isotypes, making up less than 1% of total antibodies in blood serum. The role of IgD antibodies is a bit of a mystery. We're not exactly sure what they do, but they're suspected in playing a role in reducing autoreactivity against the body's own cells and proteins, and could possibly play a role in autoimmune conditions and allergic reactions. Speaking of allergic reactions, we finally have IgE. While we mentioned IgD was in low abundance, IgE is found about 100 times less than IgD in the body. They're mainly involved in inflammatory processes, like your sinuses swelling during allergy season. The 1-2 punch of the Fab binding to the antigen and the many versatile isotypes of FCs calling in responses are what make antibodies such a powerful immune defense. Well, we can see that you're tired. We'll leave it there for now. But we'll see you in the lab soon. Thanks for watching our first entry on our Antibodies 101 series. Don't forget to subscribe to the Addgene channel to catch our latest videos. And if you're in the sharing mood, click share to help spread the word. Need more antibody info? Check out Addgene's antibody guide and blog. Addgene, a better way to share science.